Dr. Michael Hamblin, the world's leading authority on photobiomodulation, associate professor of dermatology at Harvard Medical School, and now a distinguished visiting professor at the University of Johannesburg.
Photobiomodulation is affecting the oxygenation, the mitochondria.
Do you believe in the influence of PBM on improving intestinal blood flow?
Remember, the light is scattering a lot, so the idea of a beam getting down to the intestines, I don't think it's realistic, actually.
So today it's my privilege to introduce our speaker and scientific advisor, Dr. Michael Hamblin, the world's leading authority on photobiomodulation. Dr. Hamblin was an associate professor of dermatology at Harvard Medical School and now a distinguished visiting professor at the University of Johannesburg. He has also been a visiting professor at a number of other institutions. His extensive research portfolio covers areas such as photodynamic therapy, photobiomodulation, drug delivery, nanomedicine, and tissue engineering. With over 840 peer-reviewed publications and 10 patents, Dr. Hamblin's contributions to science are both prolific and impactful. He serves as editor-in-chief of Photobiomodulation, Photomedicine, and Laser Surgery. His work has earned him an impressive H-factor, H-index of 166, with more than 116,000 citations. And this is incredibly high measures and reflect years of influential work. Dr. Hamblin's contributions have been recognized through numerous awards, including being named a fellow of SPIE, which is the International Society for Optics and Photonics, receiving the first NAALT Andre Mester Lifetime Achievement Award, and that's the North American Association for Photobiomodulation Therapy, the Dose Response Society Outstanding Career Award, the inaugural Walt Ali Award. WALT is the World Association for Photobiomodulation Therapy, and the European Medical Laser Association Lifetime Achievement Award. So please join me in welcoming Dr. Hamblin as he speaks about the history and mechanisms of PBM with a focus on treatment of brain disorders.
Thank you for the introduction. This afternoon, I'm going to tell you a bit about the history as well as the mechanisms of PBM and a bit about its applications for brain disorders. I thought I'd discuss the history because it's not very well discussed in presentations. So believe it or not, we can even go back 2 million years ago to the early stages of human evolution, where there was a hypothesis that when sort of apes, our ancestors, lost their hair, it allowed natural PBM to take place. And it could explain the rapid expansion of the human genus brain. Again, it's a theory. There's no hard evidence for that, but it is 2 million years ago. And in ancient Egypt, India, and China, there are great traditions of traditional medicine going back thousands of years. And probably the one that most concentrates on light is ancient Egypt, where they had a sun god, and the disciples used to expose themselves to the sun because they were worshipping their god, but they also did it to treat numerous diseases. In some cases, added natural herbs and natural products to go with it. Getting a bit later, Rathus and Draper, they were the first ones to actually described the first law of photochemistry, which states that light must be absorbed by a chemical substance in order for any reaction to take place. This is still important today in photobiomodulation because people are searching for chromophores and photoreceptors. Arnold Rickley in Bled in Slovenia, he was a great proponent of what today might be naturopathy. In other words, going outdoors for health benefits and for healing diseases. And he, a great proponent of sunlight. Water is good, air is better, but light is best of all. Theobald Adrian Palm trained in medicine in Edinburgh in Scotland. And as part of his training, he spent a period in Japan, in a particular sunny area of Japan. And he noticed that the children there did not have any rickets, whereas in Edinburgh, which was old reeky with a pall of smoke, the children were notorious for having rickets. So he figured out that the big difference was the sunlight. And although he didn't personally discover vitamin D, he was certainly the first one to show that light could prevent rickets, and it was subsequently found to cause the synthesis of vitamin D. Everybody has heard of Niels Ryberg Finsen in Denmark. He actually won the third ever Nobel Prize in physiology and medicine in 1903. And you'll notice he died at the young age of 44 because he had an inherited disease. In his Nobel lecture, he said, "My disease, Niemann-Pick, played a very great role in development, but he began to believe that his condition would be helped if he spent more time in the sun." So this motivated him to really do two major types of phototherapy. One was to treat cutaneous tuberculosis. The second was to treat smallpox. So cutaneous tuberculosis, he used what he thought was UV light, but was actually blue-violet light. And to treat smallpox, he used red light. And he had a Finsen lamp, and he could dye. You had filters to get either the blue, violet, or the red light onto the patients. And as I say, the third ever Nobel Prize. This triggered a lot of interest in light therapy. There were books written about it, and it depends whereabouts in the world you were. If you were in a very sunny part of the world, you could use sunshine, but some parts of the world were not very sunny, and they actually could use electric lights because Edison invented the electric light bulb a few years before, and John Harvey Kellogg in Michigan, and not a very sunny part of the USA, was a great one for electric light bars. And he exhibited them in like the World Trade Exhibition, and they really caught on ways to expose people to light for all sorts of diseases. So Oscar Bernhardt, Swiss gentleman, he was one of the first ones to really go after heliotherapy, which is the therapeutic use of sunlight. And he built a clinic in Switzerland where he could expose people to the sun in the clear mountain air. Auguste Rollier, again, another Swiss, was another proponent of heliotherapy. And you see all these little kiddies out in the sunshine, even in the snow, because up in the mountains, Rollier thought that the temperature in the mountains was important. In other words, he didn't want hot air bath, he wanted a sun bath. But my hypothesis, it's the thin mountain air that's important because the low oxygen partial pressure changes the mitochondrial metabolism. So Albert Einstein had first proposed the phenomenon of stimulated emission and population inversion back in 1916. But it took another 44 years before Ted Mayman could actually build the first working laser. There was a lot of people trying to build a laser, but Ted Mayman was the first one to actually achieve it with a tiny little thing, a flash lamp pumped ruby laser. Everybody thinks it would be some great mountainous beast, but it was just a tiny little thing. In 1963 in Boston, Paul McGuff got a ruby laser, quite a powerful ruby laser, and he shone this ruby laser beam onto tumors, first of all in rats, and secondly, he did some human patients, and he found with a powerful laser beam, you could actually cure a tumor. A few years later, 1965, Andre Mester in Budapest developed an interest in laser research, and he also got a ruby laser. We attempted to repeat Paul McGuff's anti-tumor laser treatment, but his laser had nothing like the power of McGuff's laser, so it was insufficiently powerful to cure the tumor. And then he thought, I've got this laser, let's see if we could cause skin cancer in mice. But this failed as well. So he couldn't cure cancer, he couldn't cause cancer, but he did observe increased hair growth and better wound healing in his mice and rats. And this was a fairly major discovery back in the day. And the first paper, I think, was in 19... And he went on to publish a lot of papers on this, what became known as laser therapy, low-level laser therapy. laser biostimulation, all these phrases that were popular back in the day. So that's the history up until low-level laser therapy. The societies that Brooke mentioned, all laser therapy societies, because people thought there was something special about lasers back then. But since then, people have realized that it's other kinds of light, LEDs in particular, just as good, which shouldn't be a surprise considering all the history I told you about electric light bulbs and sunlight. So anyway, getting on to the mechanisms, everybody agrees that the main photoreceptor for therapeutic light, red near-infrared light, is in the mitochondria, where you have the electron transport chain that whizzes round and allows ATP to be synthesized by ATP synthase. Glucose goes in, you get succinate. At this stage, the oxygen comes in and you produce protons which drive ATP synthase. So this unit complex 4, cytochrome C oxidase, is a very popular chromophore. It's not absolutely certain that it's the main or even the only or even the main chromophore, but never mind, it almost certainly is a chromophore. And the theory is that this complex protein with 13 subunits, 2 coppers and 2 hemes, so it has a lot of absorption peaks in the red and near infrared. I think it's at least 4 separate peaks, so it can absorb many wavelengths of red and near infrared light, even over 1,000 nanometers. And this is a popular theory that it's in cytochrome C oxidase is inhibited by nitric oxide, which can be photo to allow oxygen to get back in, and that's why respiration and ATP increase. Mitochondrial membrane potential goes up. You get nitric oxide released, which adds some evidence for this theory. You get a brief burst of reactive oxygen species and changes in calcium. And a lot of these mediators can do signaling cascades. So let's look at them. The ROS can activate protein kinase D, which will degrade I kappa B, allowing NF kappa B to go into the nucleus. The ATP can produce cyclic AMP, which will activate Jun FOS, which is the AP1 transcription factor. So between them, these 2 transcription factors govern the production of well over 100 separate genes. So you get genes involved in growth factors, cellular matrix, cell proliferation, and importantly, anti-apoptotic and pro-survival genes. The nitric oxide can also take part in signaling cascades. So there are several signaling cascades that can be triggered by this change in the mitochondria, and several of them will have effects that last for a long time. In other words, because you change protein production, the effects can last for hours, days, and even weeks. So we've done animal experiments that show after a single exposure to light, we get changes that last for 4 weeks. So in humans, it's probably better to have more than one exposure to light, but certainly a single exposure to light can have long-lasting effects. So this change in the mitochondria, where you switch from glycolysis that only produces 2 ATPs to oxidative phosphorylation that produces 30 ATPs. This is a big switch in the mitochondria. It has 2 important consequences. So one is the stem cells in their hypoxic niche carry out glycolysis because there's no oxygen around. When their mitochondria are activated, they leave their niche in search of oxygen and then activate proliferation and differentiation programs. So another type of cell that carries out glycolysis is macrophages with the M1 pro-inflammatory phenotype. They carry out glycolysis. But when OXPHOS is activated, they switch to the M2 phenotype, which is anti-inflammatory, and the cytokine profile changes from TNF-alpha, IL-1, IL-6 into IL-4, IL-10, and TGF-beta. Interestingly enough, the M2 macrophages or microglia carry out phagocytosis, so they could be responsible for removing amyloid plaque in the brain, which has been shown in some mouse models. There are other possible photoreceptors. One interesting one is TRPV1, which is a transient receptor potential ion channel, vanilloid type 1. It's known to be the receptor for capsaicin and chili peppers. It's also known to be activated by various wavelengths of light, blue-green light, and also by near-infrared and infrared light. And 980 nanometers was particularly effective. So it's an example of a light and heat activated ion channel. And here is the hypothesis that TRP ion channels, you've got blue-green light, which work via opsin, probably opsin 3. And that is a retinaldehyde cofactor, which switches conformation. 980 near-infrared light probably produces a conformational change by being absorbed by nanostructured water. So both these different kinds of light end up opening this calcium ion channel, allowing the calcium to go through. And the interesting thing is these TRP ion channels are in the mitochondria. So the net result is similar to what you get by activating the respiratory chain. You get ROS and more ATP. These are usually the things you measure. So you have to use things like inhibitors of ion channels to work out if it's this pathway or if it's the respiratory chain part. There's another theory that was proposed that this is ATP synthase. I mentioned that, which is unit 5 in the mitochondrial membrane. And this produces the ATP as a molecular rotor. So you see here it whizzing round. And the theory is that if the water, the nanostructured water, absorbs light, it reduces its viscosity. The rotor goes round faster and you get more ATP produced. So all of these mechanisms will end up with the production of more ATP, which is obviously what you measure. But you also get the same signaling pathways, which is ROS, nitric oxide, cyclic AMP. It's become obvious that photobiomodulation has systemic effects as well as local effects. So there must be something in the blood that can move around the body. When one part of the body is exposed to light, it can move around and deliver signals all over the body. And it was recently discovered that blood contains circulating, cell-free, respiratory competent mitochondria. So it's quite likely that these mitochondria in the blood are absorbing the light and traveling around to do their signaling in other parts of the body. So my laboratory did quite a lot of studies for photobiomodulation or traumatic brain injury in mice and rats because we had funding from the US Department of Defense. They were very interested in TBI in their war fighters in Iraq and Afghanistan at the time. So there was funding available to look at TBI. So we had various mouse models of TBI. One was simply hitting the mice on the head with a weight drop. The other was what's called controlled cortical impact, which involved drilling a hole in the skull, doing a controlled injury to the brain. And we had various ways of evaluating the neurological performance of the mice, just simply balancing and hanging on and force swim and tail suspension, all sorts of behavioral tests. We also had cognitive tests. This is the Morris Water maze, which is a test of learning and memory. And we found that using various light sources, we used a 810 nanometer laser a lot, we used LEDs, we used different wavelength lasers. So we did quite a lot of studies and found it had a remarkable improvement on TBI in mice. Even a single treatment 4 hours after the TBI made improvements that lasted for up to 4 weeks. we found by sacrificing the mice and looking inside the brains, we found several interesting things. So we upregulated BDNF, brain-derived neurotrophic factor. We upregulated synaptogenesis. Synaptogenesis is the formation of new connections between existing brain cells, happened in the cortex. And we found that we also got neuroprogenitor cells, specifically in the hippocampus, which is one of the areas of the brain noted for producing new brain cells. So we said that the light can help the brain to repair itself. It can produce these beneficial things, BDNF, synaptogenesis, and neuroprogenitor cells. Other people have found many other effects in the brain. These are the ones we concentrated on: neurotrophins, neurogenesis, and synaptogenesis. But other people have found remarkable anti-apoptotic effects, antioxidant effects, anti-inflammatory effects. Genesis increased blood vessels in damaged parts of the brain, less edema because you increase lymphatic drainage. So when brain swelling is a big problem after a head injury, increased blood flow and cerebral oxygenation. This happens pretty much in any condition. So a lot of these will apply to any condition, even things you don't actually expect, like anti-apoptosis is good for psychiatric problems, generative problems. Clearly, all these helping the brain to repair itself is important for strokes, head injuries, neurodegenerative diseases. But a lot of them apply to psychiatric and neurodevelopmental disorders, as well as sudden traumatic events like strokes and head injuries. The wide range of beneficial processes that people have documented to occur in the brains, mainly in experimental animals, because a lot of these you have to sacrifice the mouse and take the brain out to measure these things. But increased blood flow and cerebral oxygenation has been shown to occur in humans all the time. So there are a lot of devices that have been proposed to treat humans for transcranial PBM. As the years have gone on, these devices have got more sophisticated. Some of these are pretty primitive devices, Australian light buckets and various lasers and LEDs on the forehead. This was the Violite headset, various devices. These days, custom designed helmets are more common, and with one we did at Mass General some years ago. This is James Carroll's helmet. This is the Neuronic helmet. And there's other helmets. So now if you go onto the internet and search for transcranial photobiomodulation helmets, you'll get quite a wide choice that you can choose from. So to just briefly touch on some of the clinical studies we did. This was with Marnie Naser quite a few years ago now, and it was a simple 2 case reports of transcranial LED therapy with the MEDEX cluster, 870 and 633 nanometers. And basically 2 patients, both ladies, one 7-year after a car crash, had 8 weekly treatments, and her ability to do computer work improved tenfold. She got a home unit, used it daily for 5 years. Second, another lady with multiple concussions and PTSD, treated daily with a home unit, and did some memory and executive function tests, which improved by 2 standard deviations. So she was able to return to full-time work. And a lot of people who have head injuries, their life is pretty much destroyed. And this was a series of 11 patients that Marnie did, because she was quite excited by the first 2 results. And again, treated with the MEDEX clusters on the head. And you'll see out of these 11 patients here, there is 1, 2, 3 out of 11 but didn't really show any major improvements. But all the other 8 out of 11 had significant improvements that allowed them pretty much to resume their normal life. Because, as I say, the chronic effects of TBI can last the rest of your life if you're not careful. Again, this was an early study we did with Fred Schiffer on major depression anxiety. Again, it's a simple LED device on the forehead, 810 nanometers. And he did a single treatment to the forehead in 10 patients. And these are the scales for depression and anxiety. So these are the initial values. 2 weeks later, everybody had improved in both depression and anxiety. And this was after a single treatment to the forehead. And then he did another test after 4 weeks, and the improvement stayed at the same level. It was still there, but not at the same level. But remember, this is a single treatment to the forehead. And I don't think for psychiatric disorders you would expect a single treatment to be the regimen of choice. This was one Paolo Cassano did, again in the early days. This is with the 810 nanometer laser to the forehead, the same one that was used in the Phototherapy stroke trials, the same laser. So he did 6 applications twice a week for 3 weeks and followed these patients over the next 12, 10 weeks, however many. And they continued to improve after these 6 treatments. It wasn't a huge change immediately, but they continued to improve with significant p-values in this cohort of patients. He's since gone on to do quite a lot of studies for depression and anxiety, which you can look at. And he has a clinic now at Mass General Hospital where people come in and get photobiomodulation treatment for psychiatric disorders. So this is Violite Neuro. Everybody will be familiar with this. It's a headset with an intranasal LED. And we did a small clinical trial for Alzheimer's disease in Toronto, Canada. Is the eye on this trial. And these people got, I think, twice a week for 12 weeks. They got the Violite headset plus the intranasal. And this is one of them is the ADS cog, and the other is the MMSE, the mini mental state exam. This is the MMSE, that one. So this is a dramatic improvement, actually. And ADS cog goes down, that's in 3 cognition. After 12 weeks, the treatment was stopped, and then they were tested again, and they had regressed partly, not completely regressed, but partly. And so the size of this improvement was bigger than was found for the clinical trial for the drug whose name I can't remember, but it was an improvement for this drug, but not as big as this. They were given a headset and an intranasal to use at home after this. It's not recorded on these graphs, but they improved again when they used it at home. So obviously, I think for Alzheimer's and dementia, you're going to have to use it for the rest of your life. But still, important results. Marlineza is a big believer in these intrinsic brain networks, which are different parts of the brain that talk to each other through broad pathways. And the one she's really keen on is the default mode network, which is the area in the brain that's active during daydreaming. And it's not as if you want to particularly activate the default mode network with PBM. You want to balance it because it's got several areas that talk to each other. The idea is you want to get this intrinsic brain network into balance. But other networks are the central executive network and the salience network. So it's an interesting idea that you need to get these networks in the brain into balance, and that photobiomodulation can help this. And you can follow these networks with fairly sophisticated quantitative EEG, and to some degree with functional MRI can be done to sort of functional MRI study in a TBI patient. So an increased area in one part of the brain, the precuneus, which is a functional core of the default mode net. So there are a lot of common pathways in psychiatric diseases. Neuroinflammation, obviously a bad thing. Almost any brain disorder is accompanied by some kind of inflammation. Oxidative stress, again, a very common bad thing, typical of many brain disorders. It is usually after some kind of trauma, head injury, or a stroke, but it's when the calcium released from damaged cells causes neighboring brain cells to undergo death. Chondral dysfunction, again, common in multiple brain disorders, and apoptosis again. Low BDNF has been found in many conditions. That's probably the single most important factor in your brain. Impaired neurogenesis, that has been found in impaired synaptogenesis. Hippocampal shrinkage, the hippocampus is probably the single most important part of your brain, so you don't want that to shrink away. And then sort of end stage is when the whole of the cortex shrinks, and that's really bad. And to some degree, these pathological observations have been found in, as I say, many brain disorders, degenerative, psychiatric, and obviously after head injuries and strokes. So there's a lot of overlap in these conditions. Because it can promote neuroplasticity and synaptogenesis, it can treat several disorders that are characterized by abnormal wiring within the brain. So autism is a good example. Drug addiction, where these abnormal pathways get established due to exposure to addictive drugs, narcotics mainly. The addicts stop getting any pleasure and just keep taking these drugs because the pathways in the brain won't let them stop. And even if they go through withdrawal, so they're not physically addicted, they're still addicted in their brain. That's the key thing. Post-stroke rehabilitation, so to get the pathways reestablished after a stroke, clearly neuroplasticity and synaptogenesis are important. ADHD, another closely allied with autism, PTSD, obsessive compulsive disorder. So there's some evidence that all these conditions, disrupted neuroplasticity, can be helped by photobiomodulation that can stimulate the rewiring of the brain. Children seem to respond very well to photobiomodulation for autism, possibly because their brain is intrinsically more plastic because it's growing. I think that autism especially is going to be a rapid growth in applications, as I say, because it's often diagnosed in children, and children seem to respond particularly well. Addiction is a disease of neuroplasticity. As I said, detox is only temporary. The dopamine circuits in the brain are permanently activated, and brain imaging studies show that the brain, even when cues are shown to addicts who've been free of drugs for at least a month. So the idea is you need to rewire the brain to completely get off drug addiction. Fred Schiffer, the one that did the first depression study, he's famous for this idea the brain can be separated into two halves, and you can figure out which half is important in the individual with his patented glasses, black and white glasses. So when he treats people with photobiomodulation now, he will use his glasses to figure out which is the important side of the head in that individual. But he treated drug addicts, got a very large effect size, unilateral transcranial photobiomodulation, significant p-value in a 17 in each group. So not a huge number getting a positive p-value. He's also continued to do studies for drug addiction. with his unilateral photobiomodulation approach. This is Barney again. So people that have a stroke quite often can't speak. They have aphasia. And again, Barney believes treating the right side of the head for aphasia is important. So it's not a matter of putting a helmet on the whole head. You have to treat the correct side of the head. But she looks at functional connectivity within these intrinsic neural networks. And we did a study with Lou Banas for stroke and aphasia, just a case study, but it's additional evidence that it can help people speak again after a stroke, which is a big deal. We did one case studies or 2 case studies in ADHD, so it also helps with ADHD. The PTSD studies were really done in animals. I'm pretty sure that PTSD will work in humans, but I don't think there's been any good publications yet. But certainly in rat models of PTSD, transcranial photobiomodulation was beneficial. Paolo started to look at OCD, obsessive compulsive disorder. I think he's got a trial going for OCD, but it's the sort of thing you would expect because this idea you can rewire the brain. Bipolar disorder, again, Paolo is starting to look at bipolar disorder, which is a big deal, actually. One of the major psychiatric disorders is psychosis, even. And another, Mrs. Francisco's group in Texas, they've also looked at older bipolar patients. Now, I don't think anybody's really looked at schizophrenia yet, which would be interesting because that's the second major psychosis, of course. But many different addictions, in principle, could be treated with PBM, alcohol, nicotine, sex, internet, all these so-called addictions. Eating disorders, people die regularly of anorexia, and it lasts, and nothing can be done to get these folks eating again. So if you rewire the brain, perhaps you could get them eating again. It's certainly worth trying. And as I say, schizophrenia, it would be really interesting if that responded well to transcranial PBM. So I've separated them into these 4 groups here. Traumatic brain disorders, there are acute and chronic stroke, acute and chronic TBI, global ischemia after a heart attack. This is very common, that if your brain is deprived of oxygen for however many minutes after a heart attack, you're going to have brain damage. And similarly, in birth trauma, a lot of babies' brains are deprived of oxygen during birth. So if these folks could be treated with photobiomod relatively soon after the ischemia, perhaps the brain damage could be lessened or prevented. I've talked about all the psychiatric disorders, mental disorder. I've talked about autism and ADHD, neurodegenerative diseases, in addition to Alzheimer's and Parkinson's. There's a bunch of other neurodegenerative diseases, ALS, various dementias, CTE, Jakob, Hunt. A few folks are starting to treat patients with these diseases with photobiomodulation. So that's me done. As I say, this was my lab the last time I had a lab, which was about 5 years ago now. Thank you all for listening.
Thank you so much, Mike, for such a thorough and insightful review of the history and mechanisms and even the applications of PBM. We do have a few minutes for questions. First,
Yeah, I have one question about, is that any research done with ketogenic diet and photomodulation together on mental health care patients?
Yeah, I think there are similarities between the ketogenic diet and photobiomodulation in the effects on the mitochondria. So the ketogenic diet is quite effective in epilepsy, and people have started to propose that photobiomodulation could be helpful in epilepsy. Now, if you're asking if you would do them both at the same time,
Yes.
I don't really know the answer to that. It would be worth trying because there are similarities between the the two approaches, how they affect mitochondria, might be worth trying them at the same time.
Oh, thank you. And one more question. The metabolic syndrome in mental health patients by antipsychotic medication, can photobiomodulation helps?
This syndrome, is it to do with abnormal pathways in the brain?
No, it's due to microbiome change because antipsychotic medication change both oral and also gut microbiome.
So it changes the microbiome. Okay.
Yeah.
So the theory is the reason why photobiomodulation helps with the microbiome is it reduces systemic inflammation. It reduces neuroinflammation in the head. It will also reduce systemic inflammation, especially if you have large panels or light beds or something like that. So it will significantly reduce systemic inflammation, and that reduces or alters the composition of the gut microbiome and probably other microbiomes like the oral microbiome. The main effect is on inflammation.
Okay, thank you.
All right, JDBP.
Hi, thanks, Brooke. Dr. Hamblin, thank you for this presentation that specifically focused on the impact on cognitive function or memory when using photobiomodulation directly on the enteric nervous system over the gut.
As people talk about the gut-brain axis, which I'm sure is right, that microbiome in the gut affects the brain. Now, what I'm a bit uncertain about is whether putting light on the belly, on the abdomen, affects the gut microbiome. I think it affects systemic inflammation. So it would probably work as well to put it on the thigh muscles or the back as it would on the belly. I don't think the light is penetrating into the guts and affecting the microbiome there. It's affecting the inflammation condition, which in turn governs what microbes you have in your gut.
Okay, thank you. As an N of 1, when I put a 660-850 panel over my gut, it does increase flatulence and does reduce it does increase scale. It goes from IDSD to a proper stool. It's been very helpful in that regard.
I think it's the systemic effects that are doing it personally.
Thank you.
Guy Bauman.
Yes, Dr. Hamlin, can you comment on the potential impact of using photobiomodulation with strategies that may help to retrain neural networks such as EEG-based biofeedback or some of the neuromodulation strategies like TDCS or TACS?
No, I think there has to be similarities between other non-invasive brain stimulation techniques and DCS and RTMS and ultrasound even can all do non-invasive brain stimulation. So there has to be some similarities. I think that the time scale is a bit different. RTMS or TDCS on the head, you notice something right away, right? In general, with photobiomodulation, you don't notice a lot immediately. Maybe something, it's a slight feeling of euphoria, maybe. But there has to be similarities, but also differences. And yeah, I think that the photobiomodulation is affecting the oxygenation, the mitochondria, the blood flow, whereas the TDCS and the RTMS are creating slight neural signals in the nerves. So they're actually causing action potentials, which the photobiomodulation doesn't appear to do, at least right away. Could they be used together? I guess so. Trouble about using combination techniques together when each one is still to some degree experimental is you never know which one is playing the main role. So, but I guess they could be used together, yes.
Okay, thank you.
All right, Cliff.
Mike, lovely presentation as always. Thank you very much indeed. I've got a sort of personal question for you because I have a number of students who are on listening to you. How did you get into this field? And could you give some advice for young people, young scientists who are interested, but this wasn't their main interest, and now it is?
Yeah, so I got into this whole photobiomodulation field in a slightly indirect manner, actually. So I was trained as an organic chemist. And eventually I began working in photodynamic therapy because it involves organic compounds as photosensitizers, things like porphyrins and phyllocyanines and various fancy organic molecules. And so having worked in photodynamic therapy for a few years, use generally red light plus one of these photosensitizers. So as part of the experiments, you have to use the red light alone as a control. And what you find is that the photodynamic therapy, the red light plus the photosensitizer is killing cells, cancer cells, killing bacteria, and killing all sorts of nasty things. But the red light alone is doing exactly the opposite. It's protecting cells from dying, it's stimulating things, it's healing things, it's doing the opposite of PDT. So having discovered that the light alone controls are doing a lot, it makes sense to study it. And then gradually found it was probably more rewarding studying photobiomodulation than studying PDT. I don't think that many people in the photobiomodulation field came from basic science backgrounds in terms of, especially chemistry, a few physicists in the PBM field, but not that many chemists.
Thank you very much.
All right, Deborah McClendon.
Hi, Dr. Hamblin. Deborah McClendon from Brain Training Institute. Thank you for this presentation. I currently do a clinical model in my practice that includes a number of things: photobiomodulation, nutrigenomics, so we can look at the inflammation, neurofeedback, psychotherapy, TDCS, and also the cues. And so because I do all of this in my practice, I have to be very careful with any patients that come in with cancer. Can you share with me your thoughts about using photobiomodulation with patients who are that have gone through their chemotherapy, they don't have any cancer signs, they're in remission, or maybe in Guy's case who have had prostate, maybe they're down at like 0.01% cancer gone. What would you say about using photobiomodulation on these folks? What does the research say? And just how careful do we need to be? And do we just avoid cancer patients altogether?
No, I would say completely the opposite, actually.
Okay.
So first of all, photobiomodulation is really helpful for cancer therapy side effects, and it's possibly the single most well-accepted application now, especially in the mouth for oral mucositis and all sorts of other cancer therapy side effects, dermatitis, and many of them. So this is putting the light on the area of the body which is suffering, which is generally not where the cancer was, but not necessarily, because head and neck cancer is still treating the mouth, so in the same anatomical region. However, I don't think there's any evidence at all in humans biomodulation helps cancer to grow. It's certainly been shown in animals once or twice, but not very convincingly. On the other hand, it has been shown in a few studies that photobiomodulation in human cancer patients can be helpful in terms of their overall survival. Not being, again, studied to any major degree. I think the effects would be because it stimulates the immune system. Photobiomodulation is known to have beneficial effects on the immune system. In both autoimmune diseases, where it tends to balance the immune system, but also in stimulating it to fight infections and arguably to fight cancer, although that has not been really categorically shown. What has been shown a few times is that you treat patients with some kind of photobiomodulation. These are advanced cancer patients, and they tend to live a bit longer. So that's been shown.
Okay.
So I
Good,
wouldn't
thank
worry, I
you.
really wouldn't worry about it at all.
Okay. Okay, good. I'm still using it on those who have been cleared by their medical professional. So I feel pretty confident in that regard, but definitely want to keep an eye on the research.
I think you're going to find that modulation for cancer therapy side effects is going to rapidly increase until it's standard of care.
Great, great. Thank you for that. Thank you for that answer.
All right. Just a quick one from Gabriella. Her microphone's not working, but she's asking, when you talk about reducing systemic inflammation, she'd like to clarify the mechanism. Since we talk about systemic inflammation, we basically talk about LPS and toxins. Do you believe in the influence of PBM on enterocyte mitochondria, or in improving intestinal blood flow, or in regulating beta diversity bacteria?
Yeah, I don't know how much of the light gets to the intestines when you put it on the belly. That's the thing. Yeah, there's the skin, the muscle, what can be quite a considerable layer of fat in the belly. And then you've got the guts, which are all wrapped around each other. So I doubt that much light actually gets to the gut. Some will, but I think overlying tissue is what absorbs the light, right? The skin, the muscle, and the fat. And they all respond to photobiomodulation, and almost certainly by reducing inflammation. It's well known that the fat is a major source of systemic inflammation. And by changing the mitochondrial metabolism, you can switch cells from M1 to M2. And this applies to macrophages, but also many other cells, many other immune cells, possibly even fat cells, although I don't think that's been categorically shown.
Thank you. JD?
Yes. Last question. There are high-powered lasers that consumers are buying, like the Avant laser, 660, 850 specification. But then you've got LED panels, which are becoming quite affordable in the several $100 range, or even $100 range on Amazon. Is there much of a difference in using one versus the other? For example, depth, like to the intestines, or is it really just a matter of, for the consumer, save your money, start using it, buy the LED panel, and don't worry about the depth of a laser or the strength of a laser?
Yeah, a class 4 laser probably does penetrate a bit deeper, but I don't know whether getting to the guts, you would say if you had like a hip or a shoulder problem, you wanted a class 4 laser to get down to a joint. I think it's what's more important is the total number of joules that you put into the body. So if you have a big panel or a light bed, you can put hundreds of thousands of joules of energy in a reasonable time, half an hour sort of time, you get 100,000 joules of energy. With a class 4 laser, you're not going to do that. Even if you say, I've got this 30-watt laser, it's nothing like a 200-watt big panel, is it? In just terms of the sheer number of joules you can get in there. And remember, the light is scattering a lot. So the idea of a beam getting down to the intestines I don't think it's realistic.
All right. Thank you so much, Mike. We really appreciate the insightful overview of the history and the mechanisms and the applications of PBM. And we really appreciated all your answers, the Q&A session, and hope to have you back for a longer one in the future. Oh, I see somebody's trying to sneak in a couple of questions. Mike, do you have time for a couple of questions?
Sure.
Yeah. All right. Roberto Martinez.
Thank you for giving me this last question. Thank you for this great presentation. Very quickly, regarding lasers and laser polarization, what is the effect of the polarization?
Yeah, that's a really interesting question, because all the studies where people have tried to compare lasers with LED looked coherent, spatial coherence, and monochromatic light. But in reality, the big difference is probably polarization state. So virtually all lasers, to some degree, linear polarized. And there is some evidence that polarized light is absorbed by chromophores differently or better than non-polarized light, because the chromophores are fixed in position to some degree. And they're in a membrane, so they're not free to rotate completely at limb. So whenever you have a molecule which is fixed in position, it's likely to absorb polarized light better than non-polarized. This is it's been well established in terms of correlation spectroscopy, which looks at different polarized light. So that's well established. And there is some some devices that specifically have polarizing filters. The makers of these devices claim they perform better than non-polarized light. So it's worth considering. And LEDs generally are not polarized, but virtually all lasers are, and you can get polarizing filters. So you could fit a polarizing filter to an LED array, but I don't think anybody's done it.
Thank you very much.
All right. Then I think if there's no other questions, we'll end it for today. And thank you again, Mike. We really appreciate your time and sharing.
Thank you for having me. I've quite enjoyed